Welcome back again and welcome to Microbial Genetics. Here in this video, I will talk about Luria and Delbruck experiment and how to calculate mutation rate. We know that mutation is any change in base sequence of the DNA that is heritable. It introduced because of exposure of some chemicals and physical agents. They are called mutagens or due to the error occurred during the DNA replication by DNA polymerase itself. For example, E. coli found in the nature are sensitive to T1 bacteriophages. They infect E. coli cells and lyse or kill them. So in presence of T1, E. coli cells do not form colonies and are called T1 sensitive, and sometimes in the laboratory mutation occurred and the bacterial colonies grow. Then the bacteria are called T1 resistant. T1 resistant phenotype is stable, that means it's heritable. All progeny bacteria are T1 resistant phenotype and persist in the absence of T1. Initially, there were two hypotheses regarding how a microorganism acquired mutation. Hypothesis 1, that is called Lamarckian hypothesis. It states that characters are acquired and it's inherited. In this case, T1 resistant phenotype is induced after the exposure of bacteria to phages. So each bacterium has equal chance of survival, which is transmitted to offspring. And in hypothesis number 2, it's called Darwinian hypothesis. According to this hypothesis, T1 resistant phenotype occurs spontaneously or randomly before the exposure of bacteria to phages. Luria and Delbruck successfully distinguish between the acquired resistance and the random mutation hypothesis using statistics. If resistance were caused by exposure to the virus, then the number of resistant colonies will be same in all plates. But if resistance emerged by mutation at random times, then the number of resistant colonies will be different in different cultures, depending on how early the mutation occurred. Like in this picture, if mutation occurred in generation 1, then the number of resistant cells will be 4 after third generation. And in this picture, the red circles means mutant cells. On the other hand, if it occurs at second and third generation, then the number of resistant cells will be 2 and 1 respectively. But if mutation occurred after exposure of T1, then the number of resistant cells in all culture will be more or less same. The experiment was simple. Bacterial populations were grown up from small initial numbers to high density in a conical flask. Samples were then taken from these and grown on agar plates in the presence of phages. Several samples were taken from the same population and the numbers of resistant colonies was counted. A dilution series also performed to get the total number of bacteria per ml of culture. They also use small culture of E. coli in test tube and sampled and plated as before. When samples are taken from the same large culture, the number of colonies varied slightly between two experiments. The mean and the variance are not same. So we can conclude that random mutation hypothesis is correct. But in order to establish this hypothesis, look at the second experiment. With a large number of small culture, the total number of resistant bacteria fluctuates widely from culture to culture. In some culture, they found huge number of resistant colonies. Those plates are named as jackpot culture. You can see the variation and mean are again not equal. These findings provide a very good evidence for mutation hypothesis. They also demonstrate that despite the wild fluctuations in number among cultures, the mutation rate remained constant. They also provided method for measuring mutation rate, and therefore it is the birth of bacterial genetics. According to Luria and Delbruck, mutation rate, that is called small a, can be calculated using this equation. Here, a is the number of mutations, not mutant colonies, that have occurred in the culture, and n is the number of bacteria. They also describe how to calculate the value of small m using Poisson distribution. The probability of having zero mutation can be calculated using this equation, or simply we can say that the probability of having I number of mutations can be calculated using this equation. Now it's time to solve some numerical problem from this topic. It will help you to understand the topic better. In this example, the number of T1 resistant colonies are given in different experiment. You have to calculate the mutation rate per generation. Out of 20, only 10 tubes contains 0 mutation. So M is equal to 0.69 mutation per tubes. We know that according to Luria and Delbruck, mutation rate is number of mutations that have occurred in the culture by number of bacteria. Here, number of cells is 10 to the power 9 cell per ml and 0.1 ml of the culture is used. That means 10 to the power 8 cells. So, mutation rate is 0.69 into 10 to the power minus 8 mutations per cell division. In this problem, the bacterial cell contains 5000 genes and average mutation frequency per gene is 2 into 10 to the power 4 per generation. Then you have to calculate the average mutation per generation. Here, average mutation frequency of one gene is 2 into 10 to the power 4. So, average mutation frequency for 5000 genes will be 1 per generation. And in the last problem, 100 cultures were grown and in each culture 1 ml contains 2 into 10 to the power 9 bacteria. After incubation, the number of resistant colonies are counted and you have to calculate the mutation frequency. Here, the number of plates where no mutant colonies appeared are 20 out of 100. The probability of having zero mutation, that is called P0, is 1.6. We know that according to Luria and Delbruck, mutation rate is m by n. So mutation rate is 0.8 into 10 to the power 9. So that's all for now. If you like this video, don't forget to subscribe my channel and like, share and make comments. And thanks for watching.